Okay, we were going to pick up with this slide, light energy. I am a lazy man, and I explained this really well in a lecture video for Bio 111, so I'm sending you to John from 1 year ago. Take it away, John from 1 year ago!
Now, let's talk a little bit more about the term using light energy. What exactly does that mean? Well, light energy comes from electron excitation. That's a pretty cool term. Besides being a possible Beach Boys alternative lyrics, I'm talking about electron excitation. Woo, woo, woo. Electron excitation. Shut up. It was funny. Anyway, electron excitation is when energy is applied to an atom and causes the electrons in that atom to jump to a different orbital. Let's talk about it in terms of helium. Helium is an easy one. Helium has an atomic number of 2. So it has 2 electrons in the innermost orbit. Now, that means its valence is full. Helium is stable, it is non-reactive, and inert. Now, if we were able to apply energy to that, say electrical energy, we would cause one of the electrons in that innermost orbit to jump to the second orbit. That jump increases potential energy in that atom because this atom is now highly unstable. Not only is its innermost valence no longer full, but it has a second orbit that is nowhere near full in its valence. So this electron in the second orbit, with its increased potential energy, will now fall back down to its original orbit. And when it goes back down to its original orbit, it releases that potential energy as light and heat. Now, in photosynthetic pigments, it's not electrical energy that's causing excitation, it's light energy. In order to talk about how light causes excitation, we need to understand that light exists as waves and rays made up of photons. What is a photon? It's relatively easy. A photon is what we refer to as a fixed amount of light energy. You can sort of think of it kind of as a light molecule, but not anything really like that. It is a unit of light. See, a molecule is made of matter and has mass. But a photon is massless and there's no matter involved. It's just pure energy, but it is fixed, almost like, I don't know, a ball of energy traveling within those waves and rays. So photosynthetic pigments absorb these photons that are carrying light energy, and it's those photons, the light energy in those photons, that cause an electron to get excited. Again, it jumps to a higher orbital, which produces an increase in potential energy, and then it drops back down to its original orbit, releasing that potential energy as light and heat.
Okay, thanks, John, from 1 year ago. Let's pick this back up. So, here's a pretty cool picture. After talking all about electron excitation, I thought I'd show you that this is what happens when you have just straight chlorophyll in water, in a jar. When you shine UV light on it, it's glowing. See? This right here, this is actually light being emitted by that chlorophyll. If I were able to touch that jar, I would probably start to feel a bit warm to the touch. And that's not from the light itself. That's actually due to the chemical reaction in the pigment, due to the electron excitation. Why don't plants glow? Well, the answer is that there's a lot more to photosynthesis than just electron excitation. But because I worked really hard on it once and I wanted to show it, here's a little video that I made. It's a video metaphor. So without further ado, What just happened? Okay, so step 1 in electron excitation is that light energy is absorbed by the pigment. Step 2 is that an electron is excited by that energy to a higher orbital. When it crosses to a higher orbital, it increases both instability and potential energy in that pigment. And when it returns to its original orbital, that potential energy is released as light and heat energy, and it's more stable. So, no. Light energy, electron. Light energy is absorbed, electron to the higher orbital. It kind of works. It's a jumping dog. It's adorable. See, my dog is an electron, and this is its normal orbital. And when it absorbs energy, it jumps to its higher orbital, but it can't stay there, so it falls back down to its original orbital, the ground. So maybe that makes more sense now. Oh. So when it falls back down, energy is released as light and heat. Yeah. That's quality metaphor video. Okay, so continuing on, there are two basic stages to photosynthesis. The first stage is the light reactions, right? Guess what physical conditions in the environment you need in order for the light reactions to occur? Anyone want to just take a guess there and use your brain and think about maybe what you might need for the light reactions? If you said light, you're in fact correct. The light reactions occur while the chloroplasts are exposed to light. The light reactions consist of photosystem 2 and photosystem 1, or PS2 and PS1. Yes, I in fact did intend to write them that way. Why, you ask? Well, photosystem 2 happens first. That's right. During the course of the light reactions, light's absorbed and photosystem 2 occurs. Then once photosystem 2 finishes, the products are transferred to photosystem 1, which then finishes up. Why then is the first thing happening called photosystem 2? Because they're named in the order in which they were discovered. And when somebody walked up to the scientist who was responsible for naming photosystem 1, and said, you know, for the sake of clarity, maybe we ought to just change this one to photosystem 2. He was like, I discovered it! I discovered it first! It's mine! It's photosystem 1! And they're like, okay. Photosystem 1. You got it. And very few scientists got stabbed once they decided to keep it as photosystem 1. Okay, so light reactions occur during the daylight. The ultimate products of the light reactions are a whopping 1 ATP and a whole 1 NADPH. Amazing, huh? What do you do with these? Oh, by the way, see that? NADPH? That seem a little similar to something we talked about earlier? NAD? Yeah, I bet it does. That's because NAD was an electron carrier, right? An electron carrier. Well, guess what? NADP is an electron carrier. So, in cellular respiration, NAD was reduced to NADH. Well, in photosynthesis, NADP is reduced to NADPH. It does the same exact job as NAD, but in plants, there is no NAD. In plants, there's NADP. Well, at least in the chloroplast. So, NAD plus a phosphate is NADP+. All right. The second stage, all this was still under light reactions. So the second stage is called the Calvin cycle. Some people refer to it as the dark reactions because it doesn't need light, but that's a little confusing because you might think that they happen in the dark when really they can happen any time of day. They just don't require light in order to occur. So I don't go with dark reactions. Dark reactions dumb. I'm going with Calvin cycle. The Calvin cycle uses those products, 1 ATP and 1 NADPH, and uses them in producing sugar. Okay, so in photosystem 2, a photon is absorbed. So that's the very beginning. Light impacts a pigment, and that photon is absorbed by the pigment. And what happens when a photosynthetic pigment absorbs a photon? The electrons are excited. Specifically, 2 electrons are excited. During the process of photosystem 2, 2 water are also split. So 2 H2O are split, releasing 1 O2, because we had 2 times O, that's 2 oxygens. So those 2 oxygens are released as a single molecule of atmospheric oxygen. And then the 4 hydrogens travel to the enzyme ATP synthase. ATP synthase is that enzyme that you saw in chapter 6, right? Cellular respiration. It pumps hydrogen across the membrane, and in doing so makes ATP, or rather turns ADP and a phosphate into ATP. Now, the 4 hydrogens are sent directly to the ATP synthase. They're not carried. All right? The electron carrier is not carrying them around anymore. Now, those excited electrons from the pigment, they don't return to their original orbital. If they did, plants would glow. Plants don't glow, so something else is going on. What's going on is that they are passed into an electron transport chain. So there it is, an electron transport chain. And this is going to be very similar to the electron transport chain we saw in cellular respiration, just on a smaller scale. So the electrons from photosystem 2, those 2 electrons, pass from protein to protein in the electron transport chain. And in addition, ATP synthase is at the electron transport chain. Remember, the ATP synthase in cellular respiration was a part of the electron transport chain. Well, it just makes sense that the ATP synthase in the light reactions is also part of the electron transport chain. So during the electron transport chain, those 2 electrons excited in photosystem 2 pass from protein to protein. And the hydrogen released in photosystem 2 is pumped across ATP synthase. And what happens to those electrons and the hydrogens next? Well, remember, ATP synthase turns ADP and a phosphate into ATP, so we make 1 ATP. And then the electrons from the electron transport chain meet up with photosystem 1. And in photosystem 1, again, light is exciting pigments. So light excites pigments in photosystem 1 and releases more electrons. And so then the electrons from photosystem 2 and photosystem 1 are transferred to a second electron transport chain. That's right, there's 2 of them doing 2 different things. The second electron transport chain, instead of making ATP, instead exists to ferry the electrons over to NADP. Because remember, NADP is an electron carrier. Now, the hydrogen atoms from the electron transport chain that were pumped across ATP synthase are also transferred to NADP. And so we get NADP plus a couple electrons plus hydrogen making NADPH. And so our NADPH and our ATP are sent to the Calvin cycle. That's a lot. Feel free to pause, rewind a couple minutes, and play it again. And then ask me questions. Please ask me questions. Okay, so here we go. Here's light entering the light reactions. There's water entering the light reactions. In the light reactions, water is split and oxygen is released. The hydrogens are used to make it 1 ATP. They're pumped across ATP synthase, right? And then the electrons that are excited during the light reactions are transferred over to NADPH. So we get hydrogens and electrons transferred to NADPH. Well, NADP, which becomes NADPH, but you get the point. Anyway, ATP and NADPH now enter the Calvin cycle. The Calvin cycle is where we're going to take our carbon dioxide and our ATP and our NADPH and make sugar. Why don't we see how it's done? Okay, so here we go. Light coming in, we get the light reactions, which are photosystem 2, then an electron transport chain, which cranks out an ATP, then photosystem 1, and an electron transport chain, which cranks out hydrogen to the NADPH. So NADPH and ATP go in the Calvin cycle. Water enters and is split to form oxygen. So lots of stuff. Keep it on going. All right, so the light reactions produce a total of 1 ATP through a process called photophosphorylation. Remember, in cellular respiration, we talked about substrate level phosphorylation and oxidative phosphorylation. Well, oxidative phosphorylation used oxygen. Substrate level phosphorylation was just what occurred during glycolysis. Well, when ATP is made during photosynthesis, it's photophosphorylation. So remember, phosphorylation is adding a phosphate to something. In this case, that something is ADP, adenosine diphosphate. So when you phosphorylate adenosine diphosphate and add an extra phosphate to that, well, it now has 3, and it's adenosine triphosphate. So photo during light phosphorylation, photosynthetic ATP production. Anyway, we also get 1 NADPH. Very small in the way of products you have to memorize. Oh, I think I just spent a lot of time talking, but I want to get through this, so just deal with it. Next is the Calvin cycle. There are 3 phases to the Calvin cycle. Each round of the Calvin cycle uses 3 carbon dioxide. So 6 CO2 can power 2 cycles. Remember, because in the formula, it's 6 CO2 plus 6 H2O yields C6H12O6 plus 6 O2. So the 6 CO2 in the formula for photosynthesis can actually power 2 full rounds of the Calvin cycle. So when we say the Calvin cycle has 3 phases, what I'm saying is 1 round of the Calvin cycle is split into 3 phases. 1 round uses 3 carbon dioxide. So phase 1 is carbon fixation. And of course, the animations are out of order because I edited this and it just won't get fixed. So 3 carbon dioxide attached to 3 RUBP. RUBP is the shortening of ribulose biphosphate. Ribulose biphosphate. When you attach 3 carbon dioxide to 3 ribulose biphosphate, you form 3-rubisco. Kind of sounds like a fancy cracker, right? Or, you know, a rich cracker company. Rubisco crackers. Maybe off-brand cracker. Try Rubisco. Every bit as good as Nabisco. So anyway, 3-rubisco are then broken into 6 pieces. So phase 2 is reduction. We now in phase 2 have these 6 chunks that we broke those original 3-rubisco into. each of those 6 chunks are reduced. When you reduce something, you add hydrogen and electrons. So this is where the NADPH is rolling up. And this produces 6 G3P sugars. G3P sugars. Not glucose, G3P sugars. All right, phase 3, well, this is a cycle, which means it has to keep going over and over and over and over again. And at the beginning of the cycle, we had ribulose biphosphate. But we bound up ribulose biphosphate to carbon dioxide to make Rubisco, and then broke that into 6 molecules that became sugars, 6 G3P sugars. We no longer have ribulose biphosphate. So if we're going to be able to keep doing this, because it's a cycle, we have to get back our ribulose biphosphate. And the way we do that is to take those 5 G3P sugars and undergo a series of chemical reactions in which we convert them into ribulose biphosphate, at which point we're ready to start over again. Now, this means that of the 6 G3P sugars produced, we get to keep how many? That's right, 1. So, the total output of the Calvin cycle is 1 G3P sugar. That's not even glucose, by the way. G3P is a sugar with a phosphate attached to it. So here we go. You don't have to know the names of each single one of these or anything like that. I just am showing this to you so you see phase 1 is carbon fixation, where we take our ribulose biphosphate and attach it to 3 carbon dioxide to make Rubisco. And then in phase 2 reduction, we actually use our ATP to break this sucker into smaller pieces. So here we go. We get our Rubisco and we break it into 6 of these, and then we use ATP to further process them, and then we have NADPH to reduce them to produce G3P, and we have 6 G3P, 1 of which we actually get to keep, and the other 5, now we got to use ATP in order to send them through a few chemical reactions to make it into 1 or 3 ribulose biphosphate. So, need water? No. I'm going to get through it. So the final tally for a single cycle of the Calvin cycle is, well, our overall or our gross production is 6 G3P. However, it cost us 9 ATP and 5 G3P and 6 NADPH to produce those 6 G3P, which means our net products of the Calvin cycle are a single G3P. Those ATP and those NADPH all came from the light reactions. So you can see why the light reactions occur separately from the dark reactions, because a single round of the light reactions will not produce enough to power a single round of the dark reactions. We need 9 ATP and 6 NADPH. So you have to have a whole lot of stuff being produced in the light reactions before you can move to the Calvin cycle, the dark reaction. Anyway, so all those costs, that 9 ATP and that 6 NADPH, well, it's not like we destroy the NADPH. We release the hydrogen and it becomes NADP. And those are sent right back to the light cycle to be reduced to NADPH again. And same, it's not like we destroy ATP. When we use ATP, we hydrolyze it and break off the phosphate, and so we have ADP plus P, adenosine diphosphate plus P, and that is sent to the light cycle to be turned back into ATP through photophosphorylation. So, ADP plus P from the Calvin cycle after it's used is sent to the light cycle to be recycled, and that produces the 9 ATP, and then the NADP From the Calvin cycle, after NADPH donates its hydrogen, it becomes NADP. It's sent to the light cycle to be reduced. So that is the end of photosynthesis. May have talked a little fast, so there's no shame in rewinding here and there. Feel free. Good luck on the exam.